So today I'm going to talk about studies focusing on the evolution of SARS-CoV-2, particularly focusing on Delta, Omicron, and some newly emerging variants. So at first I briefly talk about the background. So maybe all of you know SARS-CoV-2 is the causative agent of COVID-19. And as of this May, more than 0.5 billion people were infected with SARS-CoV-2, and more than 6 million people have died of COVID-19. So meaning that SARS-CoV-2 infection and COVID-19 is an ongoing pandemic. And as all of you know, since the emergence of SARS-CoV-2 in Wuhan, this virus has highly diversified, such as Alpha, Beta, Gamma, Delta, and Omicron in over 2 years. So then from the January 2021, to promote the COVID-related researches in our country, so I have launched a consortium which is called Genotype to Phenotype Japan, G2P-Japan. And now more than 15 PIs from a variety of universities in Japan are joining this consortium to promote and accelerate COVID-related researches, particularly focusing on the newly emerging SARS-CoV-2 variants. And then actually, since January to November last year, we have published these 4 papers from our consortium. And also we have the collaboration with another consortium in United Kingdom, which is called Genotype to Phenotype UK, G2P UK. Particularly we have the collaboration with Dr. Ravi Gupta from Cambridge, and he has published 2 papers with the collaboration with our consortium, as shown here. So then I'll just briefly introduce our findings and then published in these papers. At first, we published a paper in Cell-First Microbe, and this study was mainly performed by Dr. Mototono, and then this study was performed by the collaboration of these consortium members. And in this study, we particularly focused on a mutation in spike protein L452R. And then in this study, we revealed that this mutation increases viral infectivity. And also, interestingly, this mutation evades HLA-A24 restricted cellular immunity. And then we also published a paper in New England Journal of Medicine. And this study was mainly performed by Kei, my grad student. And this study was conducted by the collaboration with these members. And in this study, we particularly focused on the Mu variant that was the prior variant of interest that was emerged in Colombia last year. And then in this study, we prepared the pseudoviruses that bear the spike protein of Mu variant, as well as the other variants such as ancestral variants, and also alpha, beta, gamma, delta, epsilon, lambda. And then we also obtained the sera from the patient and vaccinated people. And then we mixed these sera and the pseudoviruses and measured whether these variants are resistant to the humoral immunity that can be induced by infection and vaccination. And then as it's shown here, we found that Mu variant is highly resistant to the neutralizing antibodies that can be elicited by infection and vaccination. And then we published a paper in Nature, and this study was mainly conducted by Dr. Sato and Irie, and this study was performed by all of these consortium members. And in this study, we particularly focused on the Delta variant, and at first we found that the Delta variant formed a bigger syncytia when compared to ancestral and alpha and beta variants. And then we particularly focused on one mutation that can be detected in the Delta variant, P6881R. And then we inserted this mutation into the SARS-CoV-2 genome by reverse genetics, and then inoculated this virus into the viral cells, and then we could reproduce the bigger syncytia by infection, as shown here. And then we inoculated the Delta variant as well as the virus bearing P6881R mutation into hamsters, and then we demonstrated that the Delta variant and the virus bearing P6881R mutation can be pathogenic when compared to ancestral variant. And then last year, we published a paper in Cellular. This study was conducted by Izumi, my grad student, And this study was supported by these consortium members. And in this study, we particularly focused on the Lambda variant that has emerged in South American countries last year. And then we characterize the function of these mutations that can be detected in the Lambda variant. Then as shown here, by the end of November last year, in total, we published 6 papers focusing on the variant of SARS-CoV-2. And then, as you may know, in November last year, a new variant, Omicron, has emerged in South Africa. And this is the detailed history of this time. On November 25th, a new variant, B.1.6.529, has emerged in South Africa. And then South African government has announced the spread of this variant in this country. And then we, G2P-Japan, decided to focus on this variant on this day. And then in the next day, November 26th, the WHO has classified this variant as a variant of concern, and then this variant has designated to be Omicron. And then on December 7th, we obtained a clinical isolate of Omicron, actually BA.1, from National Institute of Infectious Diseases in Japan. And then we performed a variety of experiments in a few weeks, and then we could publish a preprint on Christmas. And then on the November 25th, we decided these members will join this project that focuses on Omicron. But on the next day, November 26th, Ravi has asked me to have the collaboration to focus on Omicron. So then I discussed with G2P-Japan members. Then we decided to separate our members into 2 groups. Actually, these guys will have the collaboration with Ravi to focus on the neutralization activity against Omicron. And then this team has particularly focused on the pathogenicity of Omicron. And then we conducted the 2 projects together. And then finally, we could publish 2 papers in Nature on the same issue. And then I briefly introduced our findings. The result from team neutralization that has the collaboration with Ravi. Then in this study, we found that Omicron BA.1 is highly resistant to vaccine-induced immunity. And also Omicron is completely resistant to Ronaprib. And then, as you may know, SARS-CoV-2 has 2 pathways to invade the cells. One is the temperature-dependent pathway. In this pathway, this virus has fused with the cells on the plasma membrane. But in on the other case, SARS-CoV-2 can be endocytosed after binding to S2. Then in the case of Delta variant, Delta variant prefers to temperature-dependent pathway. But in the study, we found that Omicron prefers to temperature-independent, endocytosis-dependent pathway. And in the case of the studies based on team pathogenesis, we also found that Omicron prefers to endocytosis-dependent pathway. And also we found that the spike protein of Omicron was less cleaved by furin when compared to ancestral and Delta variant spike protein. And also we found that the spike protein of Omicron is, the fusogenicity of Omicron spike protein is less fusogenic when compared to parental, ancestral or Delta variant. And also the less fusogenicity of Omicron spike protein was confirmed by immunostaining and plaque assay. And then we inoculated Omicron, Delta, and ancestral B.1.1 strain into hamsters. And then we measured the body weight as well as 2 lung parameters such as PNH and RPF. And then as shown here, in the case of B.1.1 ancestral SARS-CoV-2 and Delta variant, which is shown in orange here, these 2 variant and strains are pathogenic and body weight were decreased. But interestingly, we found that in the case of Omicron-infected hamsters, which is shown in green, so this Omicron was less pathogenic when compared to Delta and B.1.1 ancestral SARS-CoV-2. And finally, we analyzed the viral sequence deposited from these countries, and then we found that Omicron was more transmissible in the human population, actually 2 to 5-fold more transmissible than the Delta variant. And then, as you may know, a new Omicron variant BA.2 has emerged and spread all over the world. And then we should know that the difference on the spike gene between BA.1 and BA.2 is more than 2-fold more than that between Delta and Wuhan ancestral strain. So meaning that BA.2 is apparently different from BA.1, so at least in terms of viral sequence. So then at first we focused on the immune resistance of BA.2. For that we obtained 3 kinds of sera from human, hamster, and mice. In the case of human sera, we obtained 2 kinds of human sera that were infected with BA.1. And the one is the patient infected with BA.1 after 2-dose vaccination. And the other is the patient infected with BA.1 without full vaccination, meaning not vaccinated or only 1-dose vaccination. And in the case of BA.1 infected patient sera with 2-dose vaccination, we found that both BA.1 and BA.2 can be resistant at the same level. But interestingly, in the case of BA.1 infected patients without full vaccination, we found that BA.2 is more resistant to BA.1. So meaning that BA.2 is more resistant to BA.1 induced immunity. And this finding was confirmed by the study using the hamster sera, actually the BA.1 infected hamster sera. And also the resistance of BA.2 against BA.1 induced immunity was also confirmed by the murine sera that was immunized with BA.1 spike protein. And then to see the virological characters of BA.2, we prepared a chimeric recombinant SARS-CoV-2 as shown here. Actually, the backbone is the ancestral Wuhan strain, but we swapped the spike gene of this backbone with B11 ancestral spike and Delta spike, BA1 spike, or BA2 spike. And also we inserted GFP gene into ORC7A frame. And then we generated these chimeric recombinant SARS-CoV-2, and then inoculated these viruses into viral cells and we saw the level of syncytia formation. Then as shown here, we found that when compared to BA.1 spike-bearing virus, shown here, in the case of BA.2 spike-bearing virus, this virus formed a larger syncytia when compared to BA.1 spike-bearing virus. So suggesting that BA.2 spike can be more fusogenic than BA.1 spike. So to confirm that, we performed a cell-based fusion assay. For that, we prepared the cells expressing SARS-CoV-2 spike, and the other cells were expressed human S2, and then both of these 2 cells are expressed as split luciferase. So therefore, when these 2 cells are fused, we can see the luciferase activity. So based on this luciferase activity, we measure the fusion activity of the spike protein. Then as a result, as shown here, we found that the BA.2 spike is more fusogenic than BA.1 spike. Then this is the summary of our findings in the Delta variant and Omicron BA.1 variant that were published in these 2 papers. We found that these 2 parameters, spike cleavage efficacy on the x-axis and the fusogenicity of the spike protein on the y-axis are correlated to each other. Actually, this center is ancestral spike protein, but in the case of the Delta spike is more cleaved by furin when compared to ancestral B11 spike protein, and also Delta spike can be more fusogenic when compared to ancestral spike protein. But in contrast, in the case of BA.1 Omicron, as shown in the previous slide and here, we found that Omicron BA.1 spike is less cleaved by furin, and therefore this Omicron BA.1 was less fusogenic when compared to ancestral B11 strain BA.1 spike protein. So then we wondered whether BA.2 spike can be cleaved by fusion, as in the case of BA.1 spike or delta spike, because BA.2 spike was more fusogenic than BA.1. But surprisingly, we found that BA.2 spike was more fusogenic than BA.1 spike, but BA.2 spike was poorly cleaved by fusion, and the cleavage level was almost similar to BA.1. So meaning that BA.2 spike can be more fusogenic without the spike cleavage by fusion. And then finally, we focus on the pathogenicity of BA.2 spike. For that, we inoculated the chimeric viruses into the hamsters, and then we measured the body weight and two lung parameters, pH and RPF, and also we measured the level of oxygen. And in the case of, as shown in the previous slide, B11 spike protein bearing virus was pathogenic and body weight was decreased. But BA1 spike bearing virus was less pathogenic when compared to B11 spike protein bearing virus. But in the case of BA2 spike bearing virus, this virus was highly fusogenic when compared to BA1 spike bearing virus, and the pathogenicity of BA2 spike bearing virus was almost similar to B11 spike bearing virus. So meaning that BA2 spike can be more fusogenic than BA1 spike. But we should note that this virus used in study not clinical isolate. So therefore, these studies should be re-evaluated using the clinical isolate of BA2. And then we focused on the viral spread in the lung of infected hamsters. And then we performed the immunostaining on the lungs of infected hamsters. And the infected cells are showing brown here. And as shown here, in the case of BA1 spike-bearing virus, viral infected cells can be detected on the epithelial cells in the lungs, as shown here. But on the other hand, in the case of BA2 spike-bearing virus, this virus more efficiently spread in the lung of infected hamsters when compared to BA1 spike-bearing virus. And finally, we focus on the pathogenicity of BA2 spike. For that, we measure the level of inflammation by HE staining. And then again, BA1 spike-bearing virus was less pathogenic, as shown here. But we found that BA2 spike-bearing virus exhibit higher pathogenicity and inflammation in the lungs of infected hamsters than BA1 spike-bearing virus. So this is a summary of our studies focusing on Delta and Omicron BA.1 and Omicron BA.2. And here I summarize our findings on three parameters. One is spike cleavage efficacy on the x-axis and the fusogenicity of the spike protein on the y-axis. And here I show the pathogenicity of these viruses on the sides of the circles. Here, this is the center is the ancestral B1 or B11 strain. And when compared to ancestral B11 strain, we found that Delta variant spike protein was more efficiently cleaved by furin, and therefore this Delta spike was more fusogenic when compared to ancestral B.1 or B11 strain. But on the other hand, we found that Omicron BA.1 spike was less cleaved by furin, and therefore this Omicron BA.1 was less fusogenic. Therefore, this Omicron BA.1 was less pathogenic when compared to ancestral or Delta variant. But interestingly, we found that Omicron BA.2 spike was more fusogenic than BA.1, but BA.2 spike was less cleaved by furin, and the cleavage level was of BA.2 was almost similar to BA.1. So therefore, there's two remaining questions. One is how BA.2 spike exhibits higher fusogenicity without higher spike protein cleavage by furin. And the other question is, actually there's another paper from Dr. Kawako's lab as a preprint. And in this study, they showed that the pathogenicity of clinical BA.2 isolate is similar to that of BA.1 isolate. So together with our findings, now it is suggested that the non-spike region of the BA.2 genome attenuate viral pathogenicity. So then now, since January 2021, now G2P Japan has published 10 papers, and some of them has based on the collaboration with G2P UK Consortium in the United Kingdom, particularly that was led by Ravi Gupta in Cambridge. So then I'd like to thank all G2P Japan members, and also I'd like to thank my lab members shown here. Actually, I was promoted to be a full professor last month, so then this is just kind of a celebration for that in front of our building. So that's all my talk. So thank you for your kind attention, and I'm happy to have any questions.
Thank you very much, Kei, and I guess congratulations on your promotion. So again, I remind people to please raise your hands, or you can also post in the Q&A. And I'm going to go ahead and start off with a question. I'm not sure that it can be answered, but can you comment on what is the current thinking for the molecular basis for the greater fusogenicity of the BA.2 variant of Omicron?
Yeah, that's a good point. We are now doing that, and basically the higher fusogenicity can be induced by the spike protein cleavage by furin. So, but as I shown in my talk, BA.2 spike was less cleaved when compared to ancestral spike.
Yes.
So actually, we are not sure whether BA.2 spike can induce higher fusogenicity, but I think one possibility would be higher binding affinity to ACE2, because spike protein binding affinity to ACE2, BA.2 spike affinity to ACE2 is much higher than that of BA.1. So that might be one possibility, but we are not sure about that yet.
Got it. And did I understand you correctly in saying BA.2 is not more pathogenic than BA.1, and the speculation now is that that has something to do with a gene outside of the spike? Is that correct?
Right. Yes. Yeah. Actually, as shown in my talk, BA.2 spike can show higher pathogenicity, but as shown in
the hamster, correct?
So again?
In the hamster, correct?
Yes,
yeah.
right. Mm-hmm. But actually, we also obtained the clinical isolate of BA.2 and then inoculated that virus into hamsters. And actually, BA.2 clinical isolate was less pathogenic when compared to ancestral or Delta variant. So together with our findings using the chimeric viruses, I think the non-spike region of viral genome, such as ORF region or non-structured proteins, we are not sure about that, but some region can induce attenuating viral pathogenicity or less growth efficacy. So we are now doing that.
I saw that Robert Sater had his hand up, but maybe it just went down.
Steve, did you want to say something before?
I'm good.
Yeah, okay, so I had a comment. So we've done a fair amount with wild type BA.2 in hamsters and monkeys, and it's very poorly pathogenic. Hamsters don't lose weight. Monkeys have actually very low viral loads. So it's similar to BA.1 that both of them appear to be, at least in the animal models, have limited pathogenicity. And I'm not aware in South Africa whether they're seeing much in terms of hospitalization with BA.2. So there's something clearly different about this class of viruses for pathogenicity, at least in the lung. And so I think it's interesting speculation that you think this is something to do with outside of the spike gene. And I think that would be really important to understand that. And how are you planning to kind of do that going forward?
Yeah, that's a good question. Actually, now we are preparing chimeric viruses that harbors the non-spike region of BA.2 based on by reverse genetics. I mean, we are now preparing the virus that have the spike protein of B.1 virus, and then we just swapped the other regions with BA.2. So then we are now trying to identify what kind of region can cause attenuated pathogenicity.
The other thing, there's some preliminary data I saw was someone suggesting ORF6 might have a role in mediating pathogenicity. And so you might take a look at that. I mean, to see the role of ORF6 in this, because it's kind of a conundrum that these viruses are built for transmission, but not illness.
Mm-hmm. Yeah, thanks.
I was going to guess there are many, many mutations outside the spike, I'm sure, right? I mean,
Yeah.
probably dozens of mutations scattered.
Yeah.
I was going to ask, I mean, we know that the fusion assays are hugely dependent on the cell used in the assay. So I'm just thinking, oh, what is the right cell in vivo that has what level of furin and what level of ACE2? And yeah, those are very sensitive determinants of fusing assays.
That's a very good point, yes. But we confirmed that higher fusogenicity of BA.2 can be observed not only viral cells, but also the other cells such as CAR3 or A509 cells or something.
But anyway, I think it's interesting, the separation of cleavage from fusogenicity, very surprising and interesting.
Yeah, right. Yeah, we are also surprised.